Yeah, we'll start. Okay, could everybody please have a seat? And we're ready to start this morning's symposium. So this morning we have three experts in neurobiology, neuroscience, and neuroimaging. I'll introduce them all at once, and then we'll have the symposium. Barbara Franke is professor of molecular psychiatry at Radboud University Medical Center in the Netherlands. John Gabrieli, professor of neuroscience at the Massachusetts Institute of Technology. And Giedd, professor of child psychiatry at the University of California, San Diego. Each of these people have contributed substantially to our knowledge about the neuroscience of ADHD. This symposium is different from other symposia that you've attended at APSARD in two ways. One is that our goal is to address a very specific paper that was published last year in Lancet Psychiatry from the Enigma ADHD group, which Barbara will explain the results of that study, which found small but significant differences in brain size between ADHD children and non-ADHD kids, but no differences in adults. And these findings were criticized by letters to the editor as being overinterpreted as ADHD being a brain disorder. So the panel here will be discussing these issues. Barbara will present the actual results. I'll go over the critiques that were made of it. Jay and John will then talk a little bit about their views on this, and then we will open it up to a panel discussion and to discussion by the audience. The audience discussion is a little bit different because we'll have, I think, Jeff Newcorn and Joe Biederman will be carrying microphones and encouraging people in the audience to talk. If you'd like to ask a question or make a comment, raise your hand. They may ask you to make a comment if they think you know something and you're being too shy. So Edmund and Russ and people like Russ isn't shy, but I'm sure we'll get your comments. Please, this is meant to be a vocal discussion by participants. So these presentations are relatively short, and the discussion will be relatively long. So that said, we will start with Professor Franke.
Thank you very much. Also, thanks the organizers for inviting me here to give this presentation. If I can get it to start. Yes, okay. So, I'm going to show you the data that provoked this controversy about whether or not ADHD is a brain disorder. I'm presenting the work as on behalf of the Enigma ADHD working group. I will show you more about this group, and here are my disclosures and also the funding that we have for research in general and this project in particular. So we've heard a lot about brain mechanisms of ADHD already during this symposium. Joe Sargent yesterday gave a magnificent account of a number of very interesting studies that looked into brain mechanisms. If we look at the body of literature that is around and was around when we started Enigma ADHD, you find that most of the studies that have been published find differences using different neuroimaging modalities, so differences between cases with ADHD and controls, especially children, are found in different modalities of neuroimaging. But most of these studies have been really small and hampered in their power and reproducibility. Importantly, as when we started, really most of the work was done in children. There was hardly anything on adolescents and adults, and let alone old people with ADHD. So in 2014, Martine Hoogman in Nijmegen and I decided to start an ADHD working group as part of the Enigma consortium, and we approached everybody in the field who had sizable samples, and fortunately, and I was very happy to see that, all of the people really came on board and started to collaborate in this Enigma ADHD working group. So for this first analysis that we did, we had over 1,700 cases, over 1,500 controls, which is at least 3 times as large as the largest meta-analysis that had been done. So Martina decided that the first study that we were going to look at, or the first brain features that we were going to look at, were the subcortical features. That was where most of the evidence in ADHD was, and we also added intracranial volume as a measure of total brain volume. We wanted to go beyond what was already in the literature, and we decided to do that by looking at this lifespan perspective, although I have to say, and you have to remember that this is cross-sectional data, this is not longitudinal data, as Joe presented yesterday. In addition, we wanted to go beyond meta-analysis, and for that reason, we asked every site to segment their brain data again using an harmonized protocol, and we also asked for the resulting data to be shared so that we could use a mega-analysis design, so actually combine all the data instead of the meta-analysis designs that had been used before. So looking at 8 volumes, we found that indeed 6 of them were different in people with ADHD than in healthy individuals, and most of these regions had already been described in ADHD. You see the striatal structures that were smaller in ADHD, hippocampus, intracranial volume, all smaller in ADHD. The finding that was relatively new in the literature was the amygdala finding, and we actually found that this had the largest effect size. Although I have to say that looking at these Cohen's D's, this measure of effect size, all of these reductions in brain size in people with ADHD are actually small. Importantly, when we looked closer at the data, we found that most of the effects, or actually all of the effects, were situated in the children. And this is what you see here for amygdala, which had the largest effect size and the strongest persistence, I should say, when we looked into a lifespan perspective. So what you see here is age bins, so we average data over 5 years. So for example, the first age bin is 5 to 10 years, and then we move 1 year forward, so the next bin is 6 to 11 years. And so you see that, especially in the children, there is smaller volumes for the amygdala than in the healthy individuals. And the other brain regions all looked very similar to that. So we published this finding, and we were quite provocative in saying that our findings showed that ADHD was really a brain disorder, and there was a lot of resulting publicity generated and controversy generated by that. Most of the media reactions were really positive, but we'll discuss a little bit of that later. There were also some criticisms, of course. Since then, we have moved on, and we're now looking at the rest of the brain, or part of the rest of the brain in terms of the cortex. We're looking at cortical thickness and cortical surface area. And by now, we have even expanded our sample size, and we now have 36 cohorts on board. We're well over 4,000 individuals, approximately equally distributed between cases and controls. And also what you see here is that, again, the effects are in the children. There is nothing in the adults. And these are the regions that are confirmed using cross-validation. So what we did is split our sample size in half and look whether both halves actually supported this finding. And then you find that most of the findings are in the surface area measures, and only in 2 regions out of, I think, 34, there is also an effect on thickness. Again, looking at the sample, at the effect sizes, Cohen's D, small, the total surface area being the largest one, with a co-density of -25. And again, in all cases, the measures are smaller in those with ADHD than in healthy individuals. So this is a stratified analysis where we only took the children under 15 years. And this is a plot, and I apologize for the complexity, where we plotted all the areas and show you not only the children, which is the most left finding or left bar, but also the adolescents, and that is 15 to 21, and the adults, which is 22 and above. And you see that the findings are indeed strongest in the children in all cases. There is something left in the adolescents, but there's nothing anymore in the adults. If we also plot the subcortical volumes, then you see that if anything, these subcortical volumes are more persistent than the cortex, and especially the amygdala, as I already pointed out, is the strongest finding that also in the adolescents and adults. So if you do your research well, you are left with more questions than answers after having seen the results. And that was the same with us. And we started to address a number of the questions in the follow-up. The first thing, when you find small effect sizes, you ask yourself whether this is indeed true or whether there is substructure in your data, so that some of the people are normal, whereas others have relatively larger effect sizes, and if you average everything, you end up with a small effect size. We know that there is substructure in the data. You've seen the lifespan effects, so the effects are in the children, not in the adults. We checked gender effects. There's nothing there. Boys and girls are equally different between patients and controls. The question is whether there are additional subgroups, and we address that using community detection, so a way of identifying subgroups. And this is work by Ting Li, a talented PhD student in my group, who took the cohorts who had sizable numbers of children in their sample, and we looked at it for the subcortical measures. So the first thing that Ting did was to do a sort of data reduction in which we tried to bring down the number of regions to look at using exploratory factor analysis, and she came up with very reasonable factors, one of them being the basal ganglia, the other being limbic system, and then the thalamus as a loner. And then we used that data set to go on and look at potential communities within the data. And we do indeed find such communities, and they do speak to the possibility that there is indeed substructure in the data that drives the effect sizes to be relatively small. So if you look at community 1 and 2, you see that they are particularly affected in terms of their basal ganglia, but the two communities are affected in different directions, so essentially canceling the effect out more or less. And in community 3 and 4, you do not see effects. In community 3 and 4, the effects are particularly in the limbic system, and here you see the same effect. So in some cases, the regions are larger, and in some cases, the regions are smaller. So we're looking into what that means for our data, whether these communities have any clinical characteristics or are different in terms of their symptom profile or comorbidity, and that is work ongoing currently. The other thing is that we are being asked regularly is whether the effects that we see are actually the consequence of living with ADHD. Being medicated, well, we showed that medication doesn't have an effect, but living with a disorder can also have effects on your brain. And we used two approaches to look into that. And one of them was to look at families. So we had family data on about 500 individuals from the Dutch neuroimage study, in which we had patients with ADHD, unaffected siblings, and healthy unrelated individuals. And now, just to check whether you are still awake, so if ADHD was a consequence of living with a disorder, then what would the unaffected siblings look like? Would they look like controls, or would they look like ADHD cases? So who thinks they would look like controls, and who thinks they would look like controls? Who thinks they would look like ADHD affected cases? Okay. So here's the results of what we find. The unaffected individuals indeed look like ADHD, so meaning that the ADHD is unlikely to be, or the brain alterations that we see are unlikely to be a consequence of living with ADHD, because otherwise we wouldn't see them in the unaffected SIBs. They do not have ADHD, but they have the same reductions in brain size as their affected siblings have. And this is what we see throughout all of the regions that we find significantly different between cases and controls. The other way we looked at this being a consequence of having ADHD was to look into a population cohort where we look at the spectrum of symptoms with ADHD. And here we looked again at the same regions that we find affected in the case control analysis, and we do find that all of those regions are significantly associated with CBCL attention problem scales in children that are 9 to 10 years old and are part of the Generation R study, which is a large Dutch study. This is not the case for the thickness, so here there seems to be a difference. We do not find significant association with attention problems in the population for the thickness scores. So this is where we are at the moment. Just to recap, are those effects homogeneously distributed across patients? They are not, and there are some subgroups that might be interesting to follow up. And are the effects a consequence of living with a disorder? Definitely not. Otherwise, we wouldn't see them in unaffected individuals and unaffected siblings, and we wouldn't see them in the correlating with symptoms in the general population. Okay, so this is what I wanted to present. Thank you.
So my task is to summarize what the criticisms were of the Enigma results. Before I do that, it's really worth pausing to consider what an amazing accomplishment this is. What, 4,000 cases and controls, roughly, we have over multiple sites around the world. Another example of how the ADHD community cooperates to create big data to answer questions. We actually have here some real answers to the questions about brain structure in ADHD, and we shouldn't lose sight of that during the rest of this discussion. Really amazing, an amazing project, and thank you, Barbara, for leading that for ADHD Enigma. I'm going to only show one slide, and these were questions that the critics of Enigma ADHD raised, and these were raised in letters to the editor. I think there was even one group that was suggesting the article should be retracted because of the criticisms that they made. And I think the critics were from a spectrum. Some were sort of anti-psychiatry people who loved to criticize positive findings. Others were not really anti-psychiatry, but a little bit kind of, almost, let's say, anti-biology, not kind of liking papers that overemphasize the biological roots of the disorder. Really 3 points I'll mention were the questions that were raised. I think of them as questions raised by the critics. Number one, because these group differences in brain volumes are small, can we actually conclude that ADHD is a brain disorder? And we'll have some discussion about this today. What does it actually mean to have a brain disorder based upon small findings? Isn't this just normal variation and not really a disorder? Number two, if ADHD is a brain disorder, why couldn't Enigma ADHD find changes in ADHD adults? Do these findings prove that these brain changes actually cause the disorder? Number three, does concluding ADHD is a brain disorder mean that social, psychological, and environmental issues are not relevant to the etiology of the disorder? These are the questions which I'd like the audience to think about. Our panel will be talking a little bit more about that as well, and I'll probably put these questions back on the board after everybody finishes talking. But next we'll move on to Giedd to give us his impressions of Enigma ADHD and some of the questions that are raised.
Okay. Good morning, everyone. So if ADHD is a brain disorder, was one of the main conclusions, and this always sort of puzzles me that we're still fighting this battle in terms of, I'm not sure what the alternatives are, right? So I had a nightmare last night. We have the Scientologists on one side, and we'd have like the spleen people on the other, because what is the other option in terms of, and so it's odd that this is a legitimate battle that that we're still fighting. But if we say that it is a brain disorder, the obvious question, like, in what way, in what features, and so a lot of people in SUNY and stuff during our careers, MRI became available, no radiation, you can scan children, and even over time. So that was the kind of the theme for a lot of us, was let's see what this disorder is. I'm also going to just touch really quickly on this idea of delay in maturation, and I think an important nuance of that, and then hopefully we can discuss a bit the surprising finding that the amygdala was one of the bigger effects. So this idea of disorder of the brain, the evolution of exploring this has gone through 4 general phases, and the first was like, let's find a hole in the brain. So if people have schizophrenia, autism, ADHD, so let's do an MRI scan, find an abnormality, and that sort of largely failed. So in over 200 ADHD scans longitudinally over the NIH, they had no more incidents of abnormal findings in the control group. So we don't really have this idea of a true, quote, hole in the brain. But it's very ingrained in our culture, in the legal system, a lot of it emanating from Charles Whitman, who was Time Magazine Man of the Year. In 1966, climbed the Texas A&M Tower, shot 14 people after having killed his mom and people on the way. And then at autopsy, they found that he had a lesion that people often say it's in the amygdala. It was actually a very gross region that hit on a lot of areas, but this has really become a big precedent now. Let's look for these hole in the brain. In the legal system, there's still this idea of, is it you or your brain who are doing that? But it was largely unsuccessful. Almost everything I'm going to say is for ADHD and for everything else in child psychiatry in terms of not of diagnostic utility. The next phase was looking for group average differences in size. So radiologist says, oh, that's a normal scan, but different parts are bigger. Enormous human variation, right? And in the brain, it's about like this. Actually, the closest physical thing to brain variance is shoe size. In terms of, you can see people of same age and stuff, about 2-fold difference, kind of thing. But this is where a lot of the generation of studies did. You'd take 2 groups, you'd split them, and you'd look for these group average differences. And in this example, there's huge overlap, as there is in the Enigma study as well. So if you look at the actual distributions, they're much more alike than different. The next phase was then to look not just at snapshots in time, but how does the brain change through development. And this allowed us to better tell IQ, male from female, heritability turns out to be very age-dependent, separating health from illness. So that's one of my, I don't know if like a critique of the Enigma study because it's, it'll get there. But in a sense, looking at group average size differences in one-by-one nodes is 15 years behind sort of the current thinking, which is now neural networks. And all these sort of are on top of each other. They don't sort of replace, but we're looking at how the brain is connected amongst itself over the course of development. And it's much more powerful in separating like schizophrenia from even things like IQ, looking at how two areas of the brain go together. So when I see these studies now about kind of the chunkology, this part of the brain and that part of the brain, I'm really surprised that we don't find robust findings in single, the size of single things. So this was a story from our lab with Javier Castellanos and Philip Schott at NIH, this idea that ADHD is delay of maturation. And I think this was misinterpreted in sort of, oh, just wait it out. If there's a 3-4 year delay, pretty soon people will catch out, just sort of get through those tough years. And I think that completely missed the point. And this is a theme that we see a lot. This is looking at IQ with very high, medium high, and average. And what we see from these different trajectories is that if you look at age 19, can't tell you anything. Right? They're the same. It's only when you run the clock backwards that you see the separation. And in fact, the later maturing was the high IQ group. But this is the analogy I was trying to think of this morning, kind of like what would be a way to think of this. So let's say you have a college, so UCSD. We have room for 10,000 students. Okay? So one year we're going to get 20,000 applicants. We'll get a certain SAT, certain GPA, certain averages. Okay? The next year we have 50,000. Right? There's still only room for 10,000 students, but it's more competitive. The more applicants we have, then all the averages will go up, all the other things. And so there are certain constraints to how big a brain can be in adult size, largely driven by energy, minimization, and those factors. And so having a delay isn't just sort of shifting things over. The path matters. And I think the path matters a lot. And so when we talk about a delay in maturation, I think it's maybe people might misunderstand that as truly just shifting things over, because there are a lot of constraints in the adult brain. And so having more competitors, in a sense, can affect it. And autism might be the opposite, too many competitors. Their brains are larger. So those are the point I wanted to make about delayed maturation and why we quite commonly see greater effects in childhood than adult. And maybe just to kind of prime us for the amygdala, my favorite Batman character, but this truly was a surprise because there's lots of data from primates, from lesion studies that this is largely a frontal basal ganglia cerebellar circuitry. And so this is, I was very intrigued at this notion of the amygdala being one of the big differences. And an aspect of this is we call it the amygdala, and so it has at least these 7 very distinct subcomponents, all different afferents and efferents. It almost makes no conceptual sense to call it one thing. It's sort of sharing an apartment with completely different things. And the amygdala it's hard enough to measure as it is. So we have this kind of heterogeneity and sort of kind of big chunks. We have, hyperactive, impulsive, but even just inherently, so the diagnosis, we have 6 of 9 of this symptom and 6 of 9 of that. So anybody know how many combinations that comes out to? So, I just did 116,220, right? So that just inherently in the diagnosis, there's a lot of variations on this theme. So I think you can have ADHD with a frontal hit. You can have ADHD with a basal ganglia hit or with a cerebellum hit. Every link in the chain has to work. And any of them will lead to the final pathway. So I think these are really challenges that we have. Definitely not sort of slamming the study in the way. It's just the best we've done. But I think that we have a long way to go in terms of improving how we can really look at what is different in the brain.
John. Lost the mouse. There we go.
Thank you. It's a pleasure to serve on this panel of leading thinkers in this field and leading discoverers. So as a neuroscientist, we believe as a matter of faith that variation in behavior depends on variation in the brain. And that if we're not getting that right, it's because our understanding of behavior and our understanding of the brain is just so modest, both conceptually and in terms of how we're able to measure it. So we're not going to go back to look at the lungs, kidneys, and livers. We're not going to think that, and it doesn't have to be, by the way, in terms of a psychiatric diagnosis. We see brain differences in all kinds of ways in cultures and all kinds of different ways. So any behavioral difference, we believe, must ultimately be understood as a difference in the brain. And so if we don't get it, it's because we're not measuring it right or we don't have the right conceptual framework. That's my disclosure. So this is what we all dreamed of when we started doing neuroimaging in this field. We create bimodal distributions in studies of patient populations, right? That patients have a certain set of behaviors that are atypical, and then we go find controls, and typically we'll be very careful that the controls are not even close, right? Except for family studies or things like that. But, so we create this huge disparity between the control group and the patient group on the dimension of behavior that we're interested in. And then we would expect, with the right kind of neuroscience or brain imaging, that we should get something like that in the brain. And as Giedd pointed out, when you talk about something complicated like attention or control and regulation of impulses, there can be many different reasons why things end up differently. But still, we expect, we would have dreamt of this sort of match between behavior differentiation being somewhat matched up in terms of neural differences in structure or function. And yet, Jay showed you that scatter plot. We never come close to that, right? So this is not a point that we like to emphasize. You don't get points for grants or papers to go, yeah, we got a difference, but it was so tiny, and most of the individual patients could not be characterized in that way, and that's not quite fair. It has to be most, but it could be, 50%-ish, 60%-ish, and the other 40 who are completely legitimately having difficulties, fall into the same distribution in many ways as the controls. And so, we don't emphasize that point, but that's just where the field is, and it's not just in ADHD. So what are our sources of problems? There could be many. One of them is we just have a huge measurement problem at many different levels. So I especially work on functional MRI. The signal we have of the bold signal, as anybody who practices it knows, is terrible. Okay? In terms of anatomy, Giedd pointed out, for example, that the nuclei within the amygdala are distinct neurobiological entities. Animal researchers will study one nucleus compared to the others, and we mostly have to blend that in one average. So the very beautiful work from Barbara and her colleagues about the amygdala, it could be one nucleus in there, it could be a smoking gun, right? And we would have a very hard time seeing that, even the best of the results. And that's just the field. Second one is, again, we've just touched on the previous talk, that we might be very bad at identifying the consequences of complex developmental changes over time. So maybe longitudinal studies help us, and you saw some nice examples of that. But it might be that the subtlety of developmental differences is not well suited to the simplicity of neuroimaging that we have. And the third one, and this was touched upon too earlier, is that neurobiology within a diagnosis may be even more heterogeneous than we sort of already suspect it is. That it may be not just a little bit or somewhat, but stunningly so. And more than we might have imagined. So this is one example where we think, from work with Joe Biederman and his colleagues, Aaron Matfeld, where we divided up individuals who are controls, that's a scatter plot on the left, each diamond, on a working memory measure, a spatial working memory measure. On the right are adults with ADHD. The adults are the ones who fail to show the anatomic differences, right? And we saw this huge range of performance in working memory, so we simply divided up people with adult ADHD into the group that did well on this complex working memory task and the group that did not. So we're dividing up by some sort of behavior or phenotype within ADHD adults. And we gave them another test of working memory, I'll just say this briefly. And the arrows on the bottom show you that the same group that was impaired on spatial working memory was impaired on this other measure of complex working memory. So we think it's reliable, but only half of the patients have that. And the distribution that for any given measure of executive function, only about half of ADHD patients will fail a measure. Biederman and his colleagues have shown that before, as have many other researchers. So we say there's a difficulty in executive function, but any given measure, only about half of patients will show that in the laboratory. And then when you look in the brain, we find exactly the corresponding finding. So here's activation while these individuals are performing a complex working memory task. The controls on the left are activating the classic executive function areas of bilateral frontal, dorsolateral frontal areas, and parietal areas. The patients with unimpaired behavioral performance on this task, their brain activation is just like that of the controls. Their behavior is like that, and their brain activation is like that. And then the half of the patients who were bad on working memory measures, they showed the reduced activation. If we had simply averaged across these groups, we would have gotten no statistical significance difference, right? Because if half are just like controls and half are less, we'd have a small trend, right? If we had 1,000 people like that, we'd pull those things apart. But if we had just averaged those, and by the way, in the literature, when you look at this task, a fascinating thing is a lot of papers will say, oh, we get a behavioral difference but not a brain difference but not a behavioral difference. We don't know this for sure. It's easy to look back at other papers and have guesses, but there's a good chance that's because you averaged across fundamentally different people on this dimension of ADHD. And so how deep does that phenotyping have to go? And how, and even our ideas about working memory are pretty loose in many senses. People define them differently, measure it differently, and so on. So, it's the constructs that are also being developed in parallel as well as the brain measures. So returning to speculations, I think all of these are factors, the developmental issues, the measurement issues, but I think the biggest one is, my guess is, that the stunning heterogeneity across patients who share clinical needs and clinical features. So it's extremely rare to get non-overlapping brain measures. And just to put ADHD in the context of some other areas that I work or read about, you get more of a separation in patients with many years of schizophrenia and many years of treatment, but practically nobody gets non-overlapping data on brain measures, even in the severity of schizophrenia. Right? So I had a friend who works in machine learning at MIT, and she said, I'm going to take my amazing machine learning computer algorithms and separate out those schizophrenics from those controls. And she came back and said, I could do about 75% separation. All of you in this room could do 100%, right? Easily. Autism spectrum disorder, the more you work in that field, it's even worse than the story in ADHD. There is no replicable finding, on average or close to average, differences between brains of people with and without autism. Now, we defend ourselves. We say, oh, it's because we can only do imaging with people with relatively mild autism to even get them in the scanner. But there's big behavioral differences in those people with diagnosed autism. And there's no generally replicable finding on that. And if you hear the file drawer data problem in that, it's amazing. If you go give a talk on autism, people will walk up to you quietly afterwards and say, here's the 5 things that I know I'm supposed to replicate and I can't. But I won't write them up for journals because journals don't like that. So I think the field is changing. I think we're much more open to negative findings so we have an understanding of what's going on. But the autism spectrum, even more findings where there's no brain differences in children who are severe and adults who are severely affected. Dyslexia is easier. That's another field I work on. Dyslexia has a huge advantage that there's a simple, measurable behavior, which is reading a word. We don't have that in ADHD or autism, a simple behavioral measure of one thing that defines the difficulty. So I think a lot of this has to do with the complexity between everyday life that patients face and the challenges they face and that you help them with, and how hard it is to measure in the brain or psychologically specific dimensions of performance that we know how to sort of bind together between mind and brain. So these are huge challenges before us, and I suspect that when we began some years ago looking for that bimodal distribution of brain measures, we're quite shocked about the complexity of the human brain. And we only feel better because people in genetics might have it even worse. But we are more respectful. We're more respectful that the complexity that the genetics has shown, matches the complexity that we see in the measure of the brain structure and function. Thanks.
Yeah, okay. Well, you want to? Okay, so I'll open it up now for discussion by the presenters. I think I'll start first by asking Jay. You said the pathway matters, and I think that's a really important concept. I'd just like to maybe the panel to elaborate on that a little bit. So we know that ADHD brains start out different and structurally and end up, quote, typical. But are they really typical? Is it the same? If your starting point is different and your ending point is the same, is it the same brain? Is it a typical brain?
Yeah, in some ways it goes back to the idea that the trajectory is more discriminating than the single. But even in day-to-day changes, so a lot of things that affect the basal ganglia will cause inflammation and swelling. And so the autoimmune type of phenomenon. And then after that, so then it's bigger when you do the scan. And 3 days later, then it's smaller, because it's like arthritis and stuff, your joints get big. And so there's all these sort of like trajectories that are reasonably well known for some of the things, but they're usually not incorporated into the statistical models. So there's so many corrections, adjustments that you do to the data, and I think rightfully so, but each of them matters. And so one of the measures was the ICV, or the intracranial volume. And I guess the idea is that you kind of want to, like, bigger brains should have a bigger amygdala or that kind of thing. But as a pediatric neuroimager, I think it's the wrong thing to control for. It was from the aging literature, Alzheimer's, when you have a size of a skull, it's driven by brain growth. But once it starts getting smaller again, it's like a high water mark. It doesn't shrink back down. And so we have all of the data. We know how big the brain is. We have all the other measures. And so that's a correction that can really just like erase sex differences. Because the male brain is 10% larger than the female brain from birth through death. It's not height. 11-year-old girls are taller than boys. And so things like that, I think we know already in terms of that if you just sort of expand a brain, it's not like a photocopy machine. So as the brain gets bigger, you have greater percent of white matter. And it's 4/3 power law, it's well described. But it's hardly ever done in these studies. And I think that male-female differences are actually a really interesting part of this, certainly for autism. Right? It's like 7 to 8 to 1, but eating disorder 10 to 1, depression, I think is more common after. So I think that it's sort of odd that we're, I think maybe a little bit politically cautious, about looking at these male-female differences, but it really, I think, could give us insight, into these. And to do that well, then you really do need to look at these, trajectory differences. Because it's the same for males and females. The big difference is the brain maturation is earlier in females.
Yes. John, Barbara, any comments on Barbara?
Yeah, first let me say that indeed the measures that we take of the brain are very, very coarse measures. So these volumes, as you also said, they lump a lot of things together. So we're getting a very, very broad and unfocused look at the brain by looking at it that way. And in terms of looking at trajectories, I think the delay in maturation makes a big difference indeed, because you can imagine that there will be certain sensitive windows in the development of the brain that when you are delayed, like people with ADHD might be, you will not meet these. You will not have the opportunity to meet this window or to have a process going on that is restricted to this window. You will just miss the window. And that will result in a different brain. And whether that is visible at the scale that we are looking at currently is a big question mark.
Yeah, this is on, right? Okay, thanks. So, I just think the studies you described today are so beautiful and powerful in the large numbers of participants, and it's just and I know this is the inspiration for the panel. It's just amazing that what to think now about all the other studies that have been published, with smaller samples. You know, because it's hard in the field to go, you sort of emotionally, you don't want to give up on those, right? But it's hard to give them credibility, in the context of this fantastic study that you and your colleagues did. So it's a really challenging thing for us. I feel it most of all when I give lectures for teaching, which is like everything I taught about 4 years ago, we have to sort of pull back without seeming pessimistic in terms of brain basis of many things. We're small in studies. We're not as confident in what they're revealing as we used to be.
Yeah, since I invalidated my entire career, the slide, that's why I went with the sex different stuff. To totally finish me off for those controversial things.
Let me move you to talk a little bit about the question of disorder versus normal variation. This morning, Professor Thapar presented the genetic data, which is pretty compelling in suggesting that ADHD is a dimensional disorder, and as she pointed out, there are lots of other data epidemiologically that also point in that direction, that to some degree ADHD is more like hypertension, where there's a threshold that determines whether we have disorder. Is there anything in the brain data that would say is or is not consistent with that concept of ADHD being the extreme disordered version of normal variation?
Well, I guess the data that we have from the population study very, very much points in the direction that it is a trait that we're seeing throughout the entire population, and what is associated are the ADHD symptoms, and that is what causes the differences when we look at the two extremes, the controls and the patients.
I guess I'll go back to John's two-hump slide. So in terms of, I think that it's not one hump. I think it's not completely just the tail end of a normal distribution, but I'm not sure of the kind of the not equally sized humps. And I think that's really like key to understand. And for many of these things, like the windows and stuff, it's a different window for each different structure. I think there's different overlaps, at each different trait and stuff. But I think that it's such a fundamental question of is this, just a variation, extreme, or something fundamental? And a lot of the evidence in terms of not seeing brain damage would also suggest this idea that it's somewhat along a continuum for most.
Yeah, I don't think the brain. There's so much more. I have zero clinical expertise, but I don't think that changes. The brain data changes the functional needs of patients and the complexity of the gray zone. So to pick something else that's sort of simple, dyslexia. Every time we write a paper, we say it occurs in 5 to 17% of children, which seems a pretty big range. and every time you give that number, people go like, people have been working on dyslexia for many years. Can't they do better than 5 to 17? Can they just narrow it down a little bit? And the answer is, it depends on where you draw the line. It's not, some studies will say you have to be unable to read this badly, then you're at 5%. Some 7, so there is no line on this. The same thing in Alzheimer's disease. Now that we have these new methods to look at plaques and tangles quite early, right? We see plaques and tangles in lots of people who would not be diagnosed with Alzheimer's. Probably over some number of years they will be. So this idea of a sort of biological cliff that has this huge distinction between, let's say, healthy aging and Alzheimer's, every time we really measure things carefully, there's a lot of continuity, and the judgment is about functional consequences and functional support.
So there is a continuum of brain dysfunction we see in ADHD that suggests it's similar to common variation. If I can bring in another analogy from genetics, are there rare variants of people with ADHD who have more severe pathology that shows up as a rare variant, like a CNV would show up in genetics?
Well, there probably is, because we know that ADHD can also occur environmentally, as we heard this morning, but also, for example, from trauma. Trauma to the head, yes. For example, which is likely to cause trauma to the brain. Mm-hmm. So this could be something that is not continuous at that point.
I think one aspect sometimes, and if we can't draw a clear line, then it's not real. Just to follow up on your points, but lots of things in medicine are like that, right? High blood pressure, blood glucose levels, 101, you have diabetes, 99 you don't, lipid levels. Pragmatically, we draw lines, somewhere. But I think that often is part of the wide ranges of incidence of sort of, across countries and even states in the U.S., like where we draw that line. But the variance as well, so one of the very common ones is the subcortical measures that we've just been talking about. They're the only part of the brain that doesn't have a backup blood supply. And so they're vulnerable to any sort of twisting at birth, any torsion. The other parts kind of have a plan B. So there's sort of, I think, kind of a likely culprit for that in terms of some non-genetic reason. Again, subtle enough we wouldn't say, oh, here's a big hole in the brain type of thing. But I think that they're particularly vulnerable. And in the Enigma study and in my own, we often use filters to eliminate, quote, outliers. And I've wondered about that for a long time, because if it's a legitimate outlier, if it's really, a small amygdala in that person, and it's not due to a bicycle accident and stuff like that, at what point do we say, no, that's too, extreme? And so it's another one of those nuances of probably happens in genetics as well, in terms of who do you count as this homogeneous group. But so many, so birth type things, basic ganglia, head injuries, again, not getting knocked out cold, but just, soccer practice, that, is another, the frontal hit. So you can have all these different ways of breaking links in that chain. And I think that would be fruitful to know, like what, which part of the chain is broken.
John, comment, John?
No.
No more, okay. So our third question is, does concluding ADHD is a brain disorder mean that the social, psychological, and environmental issues are not relevant to the etiology? This is something that detractors of ADHD say a lot, that, the biological data are dangerous because it will make people think that the environment's not important, that psychology is not important. What do you think about that? Let's start with John.
Sure. So it's a common intuition that a brain difference is somehow genetic, and hardwired, and different from social, psychological, and environmental issues. Right? I understand why people think it's a science part of stuff, right? that goes together, genes and brains and hardware. And we all know that the way in which psychological experiences, social experiences, environmental influences affect you at one level is by changing your brain. So when we do brain measures, except maybe for those immediately at birth or before birth, which sometimes people do now, right? Even then, there's variance in healthcare and things like that, and parental stress, maternal stress. All these things show up in the brain scan. I mean, it's both an advantage and a disadvantage. We have the mix of genetics, experiences, all playing out in how the brain is structured and how it functions. And so I don't think biological evidence is for or against any kind of psychological or social interpretation. The way social things miraculously enter us is through touching our brains. And so this fight that occurs in the public, we think is terribly misguided, putting sort of biology in opposition to psychology or social factors or promoting health or positive school experiences and so on.
When I was starting in 1990, there was a paper looking at talking therapy, and for OCD versus medicines. And people made a huge commotion that talking therapy changed the brain. Right? Yeah. And that just struck me as so bizarre. Sort of like, to me, they're synonymous. Behavior is brain, brain is behavior. That if you have a behavior change, I'd be amazed if there wasn't a brain change. And it's so resilient, in terms of, you probably get it's nature and nurture still, right? Like this, you just go like, we're so, far beyond that. And I don't know how to communicate it better because it has real life implications, in terms of is it you or your brain, kind of things. And I just, I find it sort of, it's really hard to change that sort of, kind of grassroots perception that, to me, they're sort of obviously connected in inseparable ways. But I think that for some people, it has a positive effect of, oh, I'm not a bad mom. This is a real thing. It's brain-based. So I kind of get the upside of that. But I wish we could sort of move on from that basic tenet that it's a brain-based disorder.
So I guess an example of that environmental factors matter is this this example of the London cab drivers that you probably remember, that have a larger hippocampus after driving a few years or learning the maps of London by heart and things like that, and that juggling gives you more white matter in certain regions. So indeed, what we do in our environment will be reflected in the brain. And the thing that is so great about our brain is the plasticity and the ability to change and adapt. And that is what we're seeing when we're doing imaging, and nothing else and nothing less.
I'll say a little more about that while Joe and Jeff start to find people to talk about this in the audience. I've actually been criticized by someone in print who says that, Faraone just writes about the heritability of ADHD because he's trying to promulgate biological treatments, which is, of course, absurd. I've never actually even written that. As our panel has eloquently said, there's nothing in the biological theory that says a treatment has to be biological. I would encourage everybody to find an article by Paul Meehl, M-E-E-H-L, a psychologist who wrote in the 1960s. It's called On Therapeutic Nihilism. He was an early believer in the genetics of schizophrenia, even though he was a psychoanalyst and believed he should treat schizophrenia with psychoanalysis. But he has a very good paper describing why you can still use psychosocial therapies for a disorder that you believe is genetic or biological in origin. This, I think, is one of the key take-home for clinicians in terms of helping patients understand their disorder and what may or may not change it. So that said, Joe, Jeff, do you want to start distributing microphones? I'm hoping we can get Russ to talk a little bit about why adults with ADHD might be different than kids with ADHD in terms of pruning. I think you used that term in the population. And could that explain some of the brain differences?
Thank you, Steve. Thank you very much to all of you. It's a very critical issue. When thinking about the program, I was a strong advocate that we have this discussion. The ADHD continues to be a beleaguered diagnosis, so all of these things carry enormous amount of consequences. So it's very important for us, and it's very important for the way to move forward and advance the field. I also wanted to comment on the issue of conspiracy theories that accompany ADHD. Whenever there is a treatment for a condition, the condition does not exist. It's all fabrication by pharmaceutical companies. For example, when the CDC came up with statistics showing that the rate of ADHD moved from 10 to 11%, that was clearly conspirational, driven by pharma. The months later, the CDC came up with a 50% increase in the rate of autism, if you include autism spectrum, but no conspiracy theories developed because we don't treat, so we cannot treat autism very well. So the issue of being a beleaguered field is at the forefront of my preoccupation and looking at the field of ADHD. I wanted to set some ground rules. I know we have a lot of people that like to talk. So I would like to ask a question or make a comment briefly. Otherwise, like we did yesterday in the data blitz, we will use a buzzer to stop.
Just a quick comment. Thank you so much. But there's a wonderful curriculum now. Your fantastic brain that children are learning in school, and it really does help them understand how brain and behavior are linked. And I've heard children conversing with each other that one of them was hyper and getting upset, and another classmate said, "Dude, your amygdala's firing too much. Get that frontal lobe to work for you." So I think this issue of brain versus nature, nurture, brain versus behavior, all of that, we have to dispel that quickly because it continues to create these dualistic and I think fundamentally destructive opposition. So knowing that, yeah, it is my brain, and kids themselves get it. You know, they really do. So I'm hopeful.
One of the great things you were able to do at Enigma was have site investigators go back to the raw data to identify structures using a harmonized protocol. Were you able to do that at all on the phenotypic side? And this seems to be a bit of a challenge where you see results like this, and especially when you get to adulthood and so on, you know, that variation in phenotypic specification, cases and not cases, may end up being the harder thing to do, even though technologically easy data to acquire, harder data perhaps to harmonize across sites.
Yeah, so indeed we asked people to segment the data, the brain data again, using a harmonized protocol. We would have very much, or we would very much like to do the same with the phenotypic data and ask everybody to go back to their patients. That, unfortunately, is the limitation of working with legacy data. So we cannot do that, and that essentially is the border of Enigma ADHD. So we can flag up new fields to investigate, but we cannot investigate them in sufficient detail. This is something that the individual investigators and future studies should do. That is clear. So there are limitations to how the data or how the patients were assessed in terms of, no, I have to say that differently. The patients were assessed according to DSM, so all samples that we included had research diagnosis of ADHD, but used different instruments to get there, and that limits your possibilities. I showed you the data on the symptoms. This is something that we also tried in the patients and controls from Enigma ADHD and failed because there's just too much heterogeneity there. So,
I guess
Yeah.
the official subgrouping inattentive, hyperactive, and combined, did that predict things?
we haven't actually tried. We haven't tried that.
So I'm Brooke Molina from the University of Pittsburgh. Joe Biederman would like us to introduce ourselves, so that was Joe Blader, right? And my question actually is 50% of yours, but first I'd like to thank you for the large sample size and the massive work and the effect sizes. Thank you for the effect sizes, because I remember 15 years ago asking a neuroscientist, it's not what I do you ever report effect sizes? And they looked at me with the blankest face I've ever seen. So I appreciate that. One of my questions is, what with smaller samples can we accomplish to provide hypotheses to test in these larger studies? For example, we have 150 adults with ADHD histories, and I wish, for example, we had ecological momentary assessment, repeated daily assessments of all of them that we could aggregate across multiple days of measurements to capture really rich, deep clinical phenotypes that should be able to get closer to some of these very specific areas of circuitry that would relate and clarify the heterogeneity. So do you have suggestions for us with smaller samples that can be complementary to the large data studies?
You say something first. You think about what you say. Before as Barbara collects her thoughts, I don't have an answer for that, Brooke. It really is a good question. I'm not a neuroimaging person. I do know in the genetics world, I thought in the 1990s, I thought we would solve the genetics of ADHD by just having really good phenotyping and resolving heterogeneity and dividing the sample into those who had certain features and those who didn't have certain features. Published a lot about that. it turned out I was dead wrong. None of that deep phenotyping helped do anything. The only way we actually got any signal for discovering ADHD genes was to collect very, very, very large samples. And, you know, the phenotype sort of wasn't that important. Like, for example, in the study we hope will be, we expect to be published in 2018 that Anita talked about, the DeMontis et al., which you can read on bioRxiv on the web. It shows a very big correlation between a genetic correlation if ADHD is defined at, you know, in a very detailed way with structured psychiatric interviews and research centers. And you correlate that with just the clinical diagnoses in the Danish registries. So some Danish doctor said this kid has ADHD. Correlation is like 0.9. This is the genetic correlation, the correlation between their molecular genetic risk for ADHD. So in the genetics world, it's really the sample size is much more important than the phenotype. And it's, I know it sounds strange, but it's what the conclusion has been.
Yeah. That being said, it shouldn't withhold anybody to do innovative studies and try new phenotypes. And in that way, I think also the studies that are in the literature will inform Enigma in which direction to go. And this not finding any differences in the adults is something that really makes us think. So where do these differences go? Where do these changes go? Is it communication between different regions, on white matter, on functional levels? What is it? Indeed, is there any specificity for symptoms, specific symptoms in terms of what types of networks are involved? If you do studies in smaller samples, let Enigma at least guide you in terms of knowing the effect sizes that you can expect and adapting the sample sizes that you're using in your individual studies and or tempering your conclusions, being drawn on the basis of the results that you find. I think that is what I would...
Russ is going to make a comment, but I hope Jay and John will also talk about task-based MRI and more targeted investigations with smaller N samples.
Yeah, if I had a smaller N, well, 150, it almost pains me to hear that as small, because with about 30 you can find a 10% difference between groups. It depends on variation stuff, but as a rule of thumb. And with over 100, about a 5% difference. So it's actually pretty well powered, but I would try to minimize the differences in other variables. So all girls, all pre or all post-puberty, try to make them homogeneous in other dimensions, I think can increase your chance of finding differences.
I didn't know if any other comments from the panel on that, just to...
I'll just one brief one, sorry, which is that another direction that people have been doing in neuroimaging recently is incredibly in-depth imaging and with many repeated measures on a small sample. A little bit like the example you talked about with the experience monitor. So it would be, this is a guess, but if you try to make up for some of the measurement problems by bringing in a small number of very well characterized individuals and measure them 10 times in the scanner, you have more data per person than we typically do now. Yeah. It'd be interesting whether that would be helpful in this regard, whether you'd overcome some of the measurement problems by deep phenotyping and exceptional amount of data per brain. But that's a speculation.
One more comment. One more comment.
Sure, Steve.
The other comment I have is that, in genetics, we adopted, 5 years ago or so, this idea of a genome-wide significance level, and that you had to reach this very restricted level because of all the multiple testing that you do. And journals would no longer accept somebody looking at one gene and saying, I found significance in this one gene without a genome-wide correction. At least not the best journals. I think the imaging literature has caught up in the sense of, what is the brain-wide significance level of a finding? There's so many different regions people test, so many different things people can test. And I think that's why in the initial literature on, say, the brain structure and ADHD, if you go to individual studies, it's all over the map. It's only until you get a big study like Enigma that you can actually see where many of those findings go away and the real findings emerge. So I would encourage more rigor in the imaging community to define some reasonable level of significance for all the multiple testing that occurs.
All right, then. Yeah, good. For those of you who don't know me, I'm Russ Barkley. It's a pleasure to sit and listen to such great minds discuss this topic. I have two very quick comments from one who has been subjected to these criticisms for the last 40 years because of my prominence, I guess, in public speaking. But then I'll also have a question. But my first comment is this. In dealing with critics, I would strongly advise people to reverse the field and put them on the spot. What are your criteria for a brain disorder? Rather than always being on the defensive and leaving them the open field of not having to define anything, I have found that that is a devastating response to them because they have no criteria on which they have based their conclusion. So, for instance, if they say that ADHD is a myth, or it's not a disorder, or it's not a brain disorder, what is your criteria for a disorder versus a myth? What is your criteria for a brain disorder? Because clearly you must have some, since you have reached a conclusion that is contrary to mine. Share that with me. I'm not going to go first in this debate until you clarify for me what are your assumptions and what are your definitions. And frankly, it shuts them up, because they have none. The second comment has to do with the fact that there are no brain differences between the living and the immediately dead. Does this therefore mean that death is not a brain disorder? And I have used that with devastating simplicity against people, just as there is no difference in the size of the pancreas between diabetics and non-diabetics. So we're getting into here what Jay talked about, which I think is very important, and that has to do with functional structural differences and connectivities rather than just with large volumes. So I would echo that. And then I think the second comment, very quickly, is that we face the same dilemma, which I've explained to many, many reporters, as evolutionary biologists face with the theory of evolution. And that is that you are proposing scientific findings that are in direct contradiction to deeply held cultural prejudices. And obviously, in the case of evolution, that has to do with religious ideas about creation. But in the case of psychiatry and psychology, particularly in the study of biological influences, it has to do with deeply held, unquestioned assumptions about the origin of people's behavioral difficulties. And we can attribute this to Freudianism, behaviorism, and Marxism, and the success of all of these utopian philosophies about the ability to shape human behavior as we wish, and the equipotentiality and equiplasticity of it. And our predecessors, like Watson and others, have done such a great job of instilling that idea into the population's consciousness. And we are opposing that, and it becomes an uphill battle, as it is for the biologists, to convince people who have a cultural, unquestioned assumption that the origin of children's behavior problems is parenting, and specifically bad mothers, and that this can be environmentally reshaped out of these children. And even behaviorists have contributed to that idea, including some working in this field. So I tell people, look, we have a very uphill battle against these cultural biases. And just keep that in mind as you fight this fight. Biologists have been fighting this for a long time, and they're still not succeeding, at least not very well in this country. My last is a question. Why do we continue to use lab-based measures that lack validity for assessing executive functioning in individuals, and then ponder why our results turn out to be as nebulous and divergent as they are? Is this just a case of we want to look where the street light is, even though we lost our car keys in the dark? Is it because of the convenience of the test? Because the evidence that supports these tests as the gold standard for EF, as I've been championing throughout this conference, is highly questionable, if not, I think, reaching a point where the invalidity of these measures is overwhelming. And yet we continue to see people holding them up as the independent variable or the dependent variable against all other means of assessing this trait. So why do we do that?
Wow. Why do we do that? I don't do
No, first, thanks. Those are very useful, I think, insights on how to handle some of these questions that probably most of them in this room, come by. And not as a, excuse, more as a, confession. Yeah, it's puzzling. I mean, we know that like hot and cold cognition are not only different in strength, they're different circuitry, different nodes, different, edges. It's not even that we think they might be. We already know, that these differences exist at a brain circuitry level. And yeah, this is, there's people like you, I think, and others that can get this discourse rolling. I think so much of it is sometimes inertia, and as you know as well, like there's that science changes one funeral at a time, kind of thing. That's really hard to get papers accepted if you don't use a certain, well, in autism, a certain instrument to die. So that you have these sort of artificial constraints that arise, I think, from our system of getting grants, of reviewing papers, of such, that it's really hard to make these changes. I think if you had some more technologically based assessment of functioning that had more of a moment-to-moment, day-to-day activity in terms of how many times people are using autocorrect on their. You see, I think these would be possibilities to really take a step forward on capturing what we're trying to change with our treatments. But man, it's hard. It's really hard to sort of get funding, to get published if you don't play along, with instruments that have been there before, and
I suggest to allow.
Let me ask, I want to ask Russ one question. Russ, because one of the issues we face is the fact that there's no differences in adult ADHD. And in a conversation I had with you, you raised a very interesting point that maybe there are some people who don't get into studies of adult ADHD, those who go to prison, those who die young. Could you tell us your thoughts a bit about that, too?
Well, yeah, I can. We could spend hours on this, you and I, because we
2 minutes,
we
yeah.
the conversation. But I'm going to.
We also have a session.
Yes, I know.
On ADHD in the prisons.
Yeah.
Take up some of this department.
Yeah, I think that would be excellent as well. But I think the point that we make is that there is a selective culling of ADHD out of the population with time. And rather than viewing the decline in prevalence that we often see between early life, midlife, and late life that has been shown in many normative studies of ADHD rating scales, for instance, that isn't so much necessarily a developmental decline, but a removal of people from the population. Particularly more extreme cases are getting removed earlier than less extreme cases. And so that's an equally plausible explanation, not only for the population prevalence data, but also for neuroimaging data that you're losing more extreme cases as kids get older. And then, of course, there's also the issue that just because you have a normal brain size says nothing about normal brain functioning. So I would certainly want to keep both plausible ideas in mind as you look at what is happening as these people age and the differences seem to be disappearing.
Yeah. Edmund. That's true, but then still we have to keep in mind that the people that are adults in this study have ADHD. So they have a diagnosis, but they don't have these, that we don't see the same brain differences that we see in children. So, yeah.
Okay, Edmund, you're up. You're up.
Oh, I'm up, right. Okay, sorry, I was just getting, my default mode was really going. Yeah, so I want to comment on Russell. So Russell said we shouldn't be defensive, and then he gave a very defensive answer. He blamed other people's values and beliefs and presuppositions. I think we should look at our own values, beliefs, and presuppositions as well, to be honest, Russ, first of all. Secondly, this is a groundbreaking study, and I want to thank Barbara and Steve in particular for bringing all this together, because it's, I think, a field sometimes needs a shock for it to move forward. And I think this data is a bit of a shock to the field. Clearly, it's the best study, it's the largest study that we've had, and it's going to provoke, I think, hopefully a paradigm shift in the field. I mean, you've done a great job at starting to tease apart why this might be the case. Heterogeneity, a number of people have talked about that. I think that's a key issue. But also, I hope that there'll be a paradigm shift where we start to focus on development, and we start to look at the development of the brain, and we start to implement a real sort of biopsychosocial approach, very in contradiction to Russell's point of view, on the developing brain, looking at plasticity, and in particular looking at sensitive periods during development. And I think that's the crucial issue. And I suppose my question is it feasible to have a full, a thoroughgoing developmental neuroscience of ADHD across the lifespan from the earliest influences right the way through to the presentation of the disorder in childhood?
Yeah. Good question, Edmund. And a question that the field has to answer. So can we start very early with reasonable designs or just large scale and look at development? Until now in ADHD we're particularly bad at that. There are so few longitudinal studies that at all address this work. And in that way, I'm happy to see that in Enigma ADHD, at least we find in these cross-sectional studies, things that have also been seen in similar ways in longitudinal studies, so that we at least can repair this or can look at this in the absence or awaiting, I have to say, longitudinal studies that are more comprehensive than what we currently have. One thing that I want to emphasize is that this is not our study. This is a study of the field. So the study has, all of the field has contributed to that, and that is quite exceptional in imaging, I think, that all of the people, all the main players in ADHD neuroimaging get together and give their data for this work. This is really great.
I completely agree, with your thoughts on that. I mean, it's happening a little bit in terms of like the ABCD study, adolescent brain cognitive development will follow 12,000 9-year-olds through life, and just representative sampling, and some of them will develop ADHD, depression, schizophrenia, and like. Japan's doing a nationwide 8 study from birth through the first 10 years. I think it's another thing that everyone's always delighted with longitudinal studies, George Vine and stuff like that, but they don't have a strong place in the funding mechanism. Very hesitant for people to sort of guarantee a decade, of continued funding. So it's a little bit of a career gamble to do it, but I think that the world is with you on this one, that not just in behavioral health, but for diabetes and many other international type of studies are trying to accomplish what you've stated of getting well-characterized people and following them through the course of development, but development, not ending at age 10, through ages of risk. And I think it's a positive move for science.
Hi. For those of you who don't know me, I'm not Dr. Barkley. I have a comment and a question. The comment is, if we broaden out the discussion, I'm a pattern person. So 50 years ago, schizophrenia was the result of a schizophrenogenic mother, and with science, we did away with that discussion. 30 years ago, depression was a normal experience of human existence, and with science, we did away with that discussion. I anticipate we'll experience the same thing with ADHD as we gather the science. Steve humbly admitted his errors in the 1990s. It was a cul-de-sac with phenotypics, but you're now in genetics. And so in 2020, Steve, will you be telling us the cul-de-sac of genetics and what would be the next pursuit?
Oh, gosh. Well, in 2020, we won't be that, it's not that far away, David. 2020 is right around the corner. I think we'll be in a similar situation. But I think studies like Enigma, studies like the genome studies that Anita talked about, are teaching us about the nature of ADHD, about effects, for example, in the brain are small. So it means small samples are not going to be that informative unless they, for example, use the results from Enigma to test very specific hypotheses. They're not going to be very good for discovery. We know from, if you look at genomic studies, you start with a certain sample size. I mean, there's a linear relationship almost between the size of your sample and the number of genome-wide hits that you have. The same thing I'll bet will be true for brain imaging studies. If we could do 1,000 more brain images, we would then we wouldn't get bigger effect sizes, but we get more significant differences between structures in some of the structures that currently are not statistically significant. So it is, as Jay mentioned, it's the big issue about funding is how these things are funded. It's a structural problem when it comes to doing big science. Essentially, you need lots of information. You need lots of support from funding agencies.
I would like to say that there's some kind of word of caution about the conclusion of small effect sizes in light of the fact of heterogeneity most likely to be present or not. Reminds me of the instructor in my course on statistics that I tell my residents that if I eat a chicken and you eat none, on average both have half a chicken. So if 10% of your sample, the follow-up is that I'm full and you're hungry, that's a concept. But what I'm trying to say is that the conclusion that these are small effect sizes has to be tempered against the fact that we do not know if there is a group with large effect sizes that may be characterized better by future work. So I would be a little bit more cautious in the conclusion that now we need to have 20,000 brains in the next imaging study. So I think that with all the congratulations of the enormous effort that goes in doing what has been done by a large number of people, but particularly analyzing and putting the data into press, we have to be a little bit more cautious in concluding that the next study will need to have 50,000 brains because effect sizes are so small.
So maybe if I can jump in here, and as in wrapping up and getting us to our next session, it might be a good idea for a future session to figure out how to do that. What kinds of investigations with lower ends could we take? Because to recommend large-scale longitudinal studies without proximal hypotheses that'll never get funded is just not going to fly. And you know, there won't be very often another opportunity like you've had here. So we have to figure out what we're going to do. And that would be another useful think tank kind of session for us to maybe have here. I would like to congratulate the panelists. This was really a fabulous session. And I also want to congratulate Joe. This was Joe's brainchild, and it really was a brilliant idea. We've learned to listen to Joe's brilliant ideas and put them into operation in the program. And so a hearty thanks to all of you for this. I want to also encourage you to pick up some lunch and go to the discussion groups. Let the two discussion groups leaders allow a little time for the groups to coalesce. We don't expect them to go a full 2 hours. We have an outstanding session on ADHD in prisons and juvenile justice, and also an outstanding session on international perspectives on diagnosis and treatment of ADHD from people around the world.
I haven't had a chance to say hello.
Yeah, but the other thing I want to mention to you, I have to run to this other meeting I'm going to. But Andy and Johnson or Jackson, Jackson, I think. Jackson. They changed the room. They're next to each other. Tell Andy and Tony. Could you tell Andy and Tony?